Hello, this is Dr. Martin, and we're beginning our lecture on Chapter 17. We'll also do Chapter 18 in this week, which is the third week of our course. Now, remember back in previous chapters, we talked about the domains of life, and there were three basic domains that all life is classified into. Two very primitive domains of life are what we're going to cover here in Chapter 17, the bacteria and the archaea. And so let's dive right in. There's a lot of slides here we need to talk about, and I'll try to keep it as brief and as possible and as interesting as possible for you. Remember, these slides are also downloadable for you and accessible for you in this week's module. So without me talking on it, so be sure and download it as well and make your own notes as we go along if you need to. So remember, these two domains, they're very primitive. They're what we call prokaryotes. Pro meaning before. Prokaryotes are single-celled organisms. They really lack a lot of what we would consider other structures that are found in more complex organisms. They're very primitive, very small, very basic organisms. And so there's two domains here we want to talk about: the bacteria and the archaea. And then we'll talk about the eukarya in subsequent chapters after this. Look at this information on the right here. When they developed, they're very old, but look at this. No place on earth lacks prokaryotic life. It is an incredibly versatile, very broad, occupying many habitats, especially the bacteria. Not so much the archaea, but the bacteria especially. Their DNA, um, that's really how we use what we use to distinguish between these two, because they look a lot alike. But these are the two groups that we want to talk about: domain bacteria and domain archaea. A couple of questions here that you can answer, just to kind of give you a preview of what might come later as we talk about um, the upcoming exam for this particular unit, Unit 1. If we want to compare these two, it's kind of hard. It's even hard for some actual biology majors when we begin to talk about the difference between bacteria and archaea. I don't want to be too technical here because there's some terms here that we haven't talked about in the class, but prokaryotic cells at the top there in the red box, prokaryotic cells, they lack a nucleus. Remember that from your biology in high school. There's a centralized nucleus in most cells that contains DNA, chromosomes, genetic information. But prokaryotic cells are so primitive, they don't have a nucleus. They do have DNA, but it's not bound by its own membrane and enclosed in a nucleus. Very small, 1 to 10 micrometers. They kind of have a singular circular chromosome, whereas you and I have as many as 46 chromosomes in the human body. Other animals have many more. Some other features that are unique just to the bacteria: they have a cell wall, which is kind of like plants and fungi that have cell walls outside the cell. There's a big word here. The cell wall is primarily composed of a carbohydrate called peptidoglycan. We'll learn about that later. Some of them can carry on photosynthesis, but and then the archaea, which is a smaller group, very insensitive to streptomycin, which is an antibiotic. Not a whole lot to talk about here, but archaea, but we'll get to those later. But I want to spend a lot of time, especially on bacteria. They have a nucleoid region, which is where the DNA is found. Remember, it's not a nucleus. They do have DNA. They do have genes. They have ribosomes, which is found in other organisms, higher organisms as well. That's where proteins are constructed. They have plasmids that are just kind of circles of DNA that kind of float around in the cell, but a very, very primitive structure. All cells have a cell membrane. It's the boundary of the cell. It regulates a very important function, what comes into and out of a cell, the cell membrane. But the cell wall is outside of the cell, and sometimes used for shape, sometimes used for protection, but it's very distinctive. But bacteria have cell walls, fungi have cell walls, plant cells have cell walls, and but animal cells like us, we do not. Um, some bacteria have a layer of proteins that surround them. This is very interesting for science and microbiology, especially. It's what we call a glycocalyx. It helps them to attach to surfaces. It's what we call biofilms. And these biofilms help them to evade the immune system of a host organism. You have bacteria living inside of you that have biofilms that surround them, help them to attach to certain substances inside your body and hide from your immune system. It kind of keeps them safe in your body. And you need those. You need many of these bacteria that have biofilms. And some of these prokaryotes have what we call pili, which are hair-like projections. They're made of protein. Some are used to exchange DNA with other cells, like that's what's happening in these two bacteria that are end to end here. They're exchanging DNA. Very important about prokaryotes is they have a flagella, these long hair-like structures. They help them move, help them to rotate. They act like propellers. They help them respond to environmental conditions like heat or chemicals or pH. But I mean, it's similar to the flagella of sperm in human males that help them swim and move, become motile. It's very similar to what we see in human sperm cells. Bacterial cell walls are outside of the cell, very distinctive. Um, they're distinctive because they stain a certain way. Bacteria that take what we call a gram stain and turn kind of purple, the cell wall turns purple and absorbs this stain. We call them gram positive bacteria. If the cell wall rejects that stain and comes out kind of pink, we call those gram negative bacteria. I know you're thinking that's not important, but it's hugely important. When students go on and take the next class in biology, one of the first classes they take is microbiology. One of the first labs we learn in microbiology is the gram stain. It's because we use this stain to distinguish two different broad groups of bacteria, what we call gram positive bacteria, gram negative bacteria. And it all depends on whether this cell wall can absorb the gram stain or not. Gram positive, gram negative. I know you don't think that's important, but it's incredibly important in science, especially microbiology. So in the bacteria of especially bacteria here, not so much the archaea, but the bacteria, there's three common shapes. You've heard of these. There's circular shaped bacteria that we call coccus bacteria. You've heard of that. If you've ever had strep throat, then you've had a streptococcus bacterial infection. Streptococci are chains that are attached of circular bacteria. Bacillus bacteria, which is the green picture here in the front, kind of hot tamale shaped bacteria, kind of rod shaped bacteria. These are very environmental, soil, skin type of bacteria. And then there are spirillum bacteria that are spiral shaped. We learned this very early on in microbiology as well. And these are the three distinctive shapes of the cell walls of bacteria. Bacteria are classic for forming spores, what we call endospores. And again, you may think this is way too deep, but this is incredibly important. Some bacteria are known to live for thousands and thousands of years in a spore form, or kind of what we call a hibernating form. It's a loose way to explain it, but they form spores, and these spores are incredibly resistant to environmental changes, climatic changes, and Clostridium botulinum is a classic endospore forming bacteria. You know this, that if you want to purify water, you better boil it. If you want to purify or can vegetables, well, you better steam them. and heat them. If you've ever done any canning, or your mother or grandparents have done any canning, you know it's a pressure cooker that they put inside these vegetables inside of in order to purify them and sterilize them. And the reason why is endospores are so strong and so encapsulated that heat and pressure is the only thing that will kill these types of bacteria, these spores in these bacteria, incredibly resistant, except for heat and steam and pressure. That's why we use pressure cookers. Some of you probably can vegetables, and that's why is because of spore forming bacteria like this. But bacteria, prokaryotes are amazingly diverse. Um, for instance, some of them do what we call fixed bacteria. Some are heterotrophs, which means they feed on other organisms. Some are autotrophs, which means they make their own food. They're green. They live on your skin. They live in your digestive tract. They decompose, um, animal material. It's incredibly diverse. I know I use that word. I'm going to have to quit using that word, but very widely diverse, very diverse group of organisms. So let's talk about those two terms I just mentioned. What we call autotrophs. Troph means energy. Autotrophs produce their own energy. Some are photoautotrophs, that they get their energy from the sun. Some are chemoautotrophs that get their energy from chemicals, matter in the soil. But these prokaryotes are autotrophic. They produce their own food. They produce their own, especially their own carbohydrates, their own glucose. You and I feed on other things to get our energy. Autotrophs produce their other or their own energy. So heterotrophs feed on other things. They can't produce their own food, so they have to feed on other things. For instance, the bacteria that lives inside of your small intestine, well, they help break down sugars, especially. Some help break down proteins, some help break down fats, but especially the bacteria that live in your digestive system, they help to break down sugars. They're fermenters. They ferment sugars. E. coli is a fermenter inside of your intestine that helps, and they help feed on. They have to feed on other things. They rely on you to provide their food, but they're heterotrophs. Remember, troph means energy. Auto means self. Hetero means other. And these are autotrophs. Some prokaryotes are heterotrophs. Some other very important scientific terms here that we need to distinguish. Some prokaryotes are aerobic. Most organisms are aerobic, meaning they require oxygen to live. Now, you may think that sounds kind of strange. Are there organisms out there that do not require oxygen? Yes, and we'll explain that here in just a second. But aerobic prokaryotes, they require oxygen to live. Most of them live on the surface of our skin. They live in the soil. They live on your body. They live on surfaces of outdoor environmental type of conditions. These are what we call obligate aerobes. Obligate meaning they must have oxygen. Aerobic meaning they must have oxygen. But not all prokaryotes like oxygen. Oxygen is a pretty greedy element. It's an aggressive element. And we have to have certain physiological characteristics to deal with oxygen. Not all prokaryotes do. So some are what we call obligate anaerobes. Remember, I put an A in front of something and it means the opposite. So these are anaerobic prokaryotes. They don't like oxygen. In fact, they're killed by oxygen. So they only live in habitats that lack oxygen. For instance, those that live inside your digestive tract. They're kind of hidden from the environment. They don't like oxygen. There are some pathogenic organisms that can live on your skin, gangrene. can live on your extremities, and they produce their own gaseous environment that is non-oxygenated. And so that's why the decomposing smell of that kind of bacteria. So some are now. Then there are some that are facultative anaerobes, where they can kind of live in both environments. It's what we call facultative. So the difference between an obligate versus a facultative. Obligate means they can't live in an oxygen environment, but a facultative means they can kind of tolerate it. So we classify all of these prokaryotes, boy, especially the bacteria. We classify them using their DNA sequences, maybe their RNA sequences. Remember back during COVID, COVID was a virus that was an RNA virus, not a DNA virus. And same way with some of these prokaryotes, especially the bacteria, we classify them based on their DNA sequences. But sometimes we classify bacteria based on whether they have different kingdoms. We classify them into different kingdoms based on their DNA sequences. A couple more questions here. I'm going to try to move on kind of quick so we can get through this. I don't want to bore you too much, but I want to make it as interesting for you as possible. Prokaryotes, how do they reproduce? They're diverse. They live basically everywhere. They live in you. They live on you. Some you can't live without. Some you have to have. Some are very pathogenic, make you sick. So how do they procreate? How do they propagate? How do they replicate? So that's what we kind of want to talk about because it's very important. We can grow them in a petri dish, or we can grow them inside your body, grow them on your skin, grow them in your mouth, grow them on your food. So let's find out how they actually replicate. Well, some replicate what we call vertically. Now, they don't have sex cells. They don't have sperm and egg, pollen. They reproduce what we call asexually. You see the bold term here. They reproduce by what we call binary fission. It's where a parent cell, here's the chromosome, it replicates, it doubles, and then the organism simply pinches into two, and the replicated DNA separates and then we divide the rest of the cell. It's what we call binary fission. We have a parent cell, and then we have two cloned daughter cells. Both of them are identical to the parent cell. That's why we can call them technically clones. This is asexual reproduction. No sperm, no egg, no sex cells, no gametes. But we do have to replicate from 1 to 2 cells. E. coli replicates about every 20 minutes in a petri dish. 2 to 4 to 8 to 16 to 32 to 64 and then on. Every 20 minutes they replicate quickly. At that rate, in about 48 hours, it would basically spread across the globe at that rate. But we know that there are environmental conditions that press against that kind of growth and that kind of growth, and so it's going to stop it. But that's how fast these guys can replicate, from 1 to 2 cells in about 20 minutes. Now, there's not a whole lot of DNA there to replicate. That's what we call vertical binary fission replication. But some prokaryotes will transfer their DNA horizontally. There's a couple of ways to do this. There's 3 ways. On the next 3 slides, we're going to do this. There's what we call transformation, transduction, and then here conjugation. So let me explain real quick. I don't want to get too technical here, but it's important from a laboratory standpoint of what we're talking about. They can transmit their DNA by what we call transformation, where the parent cell dies, and an adjacent cell simply picks up their DNA, and then it's kind of absorbed in their DNA. This is what we call transformation. It allows scientists to do some pretty quick laboratory work. by introducing what we call recombinant DNA. We can make bacteria do certain things in the lab by this kind of replication, by this kind of reproduction, what we call transformation, where the parent cell dies. Here's another one where viruses will pick up the DNA and then be incorporated into another bacterial cell. It's what we call transduction. Now, scientists will use viruses like this to allow them to pick up the DNA and transport it to another cell, another bacterial cell. And this is what we call transductions used in laboratories. Then there's what we call conjugation. This is pretty interesting. It's pretty close to sexual reproduction in that two cells kind of snuggle up next to each other. And remember those pili we were talking about, these protein pili, they will penetrate between the cells and then they'll kind of exchange some DNA. Sounds kind of kinky, but they're not exchanging sperm or egg or pollen or anything like that, like animals and plants do. They're simply snuggling up next to each other and exchanging some DNA. And this is what we call conjugation. I know what you're thinking. Doesn't sound very important, but it's hugely important because it allows them to exchange genetic information. And from a diversity standpoint, that's very important for organisms. I'm going to fast forward through some of these clicker questions. You can look through these and some of the concepts, but I want to move on kind of quick. Just trying to identify some of the classification of bacteria and why and how we classify them. I want to talk just a little bit about some of these, but not very much. I'm going to progress pretty quick through these. I don't want to bore you too much. We're already 20 some minutes into this lecture, so I don't want to put you to sleep. So, but protobacteria, again, talking about bacteria here, gram-negative bacteria. So they're pink bacteria. These are just different types of bacteria we want to talk about. Proteobacteria, they're heterotrophic. They're facultative anaerobes that can kind of handle some oxygen, but not. They're some of them autotrophic that obtain their energy from sulfur. It's a pretty small group of bacteria, what we call proteobacteria. Here's a huge group of bacteria that we call cyanobacteria. Remember, these are greenish, bluish. Cyano means blue-green, but these are these are cells that release oxygen from photosynthesis. Very important producers in many ecosystems. Very old, probably gave rise to some chloroplasts, green chloroplasts that we see in plants, but cyanobacteria, very important in ecosystems. Spirochetes is a group here that is kind of spiral shaped. Many of these are pathogens. Lyme disease, syphilis is caused by a spirochete bacteria, but Lyme disease is a tick-borne disease, especially important in the northeastern United States and the New England states. Firmicutes are very medically important. This is a group of rod-shaped bacteria that cause anthrax, tetanus. Both of these are pathogenic on humans, pathogenic on many animals, cause skin infections, especially anthrax. Tetanus is a, I don't have time to explain it, but it's you get tetanus shots. It's a soil bacteria. We said many of the rod-shaped bacteria, bacillus bacteria, are environmental bacteria that live in the soil and on substance, but you get tetanus shots. Anytime you go with a cut to the doctor, they're going to say, when's the last time you had your tetanus shot? This is why. This is a pathogenic firmicute bacteria that lives in the soil. It can get in cuts. Treatment nowadays for tetanus is very preventative. It's a vaccine that's very preventative, but boy, it's a terrible, very rigid, full-on muscle contraction in muscles in a person who has tetanus. I've only seen photographs of somebody with tetanus where Their muscles are in full contraction, no relaxation after muscle contraction, but a very serious infection, but very preventable infection as well. Um, Moving on to the archaea. Not going to spend much time here on the archaea because there's such a small group. The bacteria, we might as well do microbiology if we're going to talk any more about the bacteria. But here's the archaea. Notice that word at the top of this slide. They are extremophiles. Phile means love. They are extremophiles. They live in oftentimes very extreme environments, like heat vents, like here. It's a prokaryote, primitive organism that lives in a very hot, sometimes very acidic environment, very toxic environment. But this is the no oxygen, very salty, but extreme environments is what we're talking about here. But they're also very essential for some cycles on land and water. Here's some groups of archaea here, Euarchaeota and some others, and I don't want to spend too much time here. But so here's two groups here. The domain bacteria, domain archaea. Clicker question number 4 here, and some more here that we can just move through. And again, a mastering comment here that we don't want to spend too much time on. So we saw this picture on a previous slide. There are these two groups of organisms: small, primitive, seemingly insignificant, invisible to the natural eye, very microscopic, not very complex at all, but very important and essential to life. One of the things we kind of want to talk about here is what we call nitrogen fixation. 79% of what you're breathing in right now is nitrogen, not oxygen. It's nitrogen. It's probably a little lower than that, about 71% nitrogen. But it's an element that you simply breathe in and breathe out. You don't use it. You can't assimilate it. You simply breathe it in and out. When you inhale, exhale, nitrogen. But you have to have other forms of nitrogen for body structure, body function, especially proteins. So there are organisms out there that take atmospheric nitrogen that you're breathing and they transform it into a usable form of nitrogen that you can ingest in your foods. This is what we call nitrogen fixation. It's where nitrogen in the air is converted to ammonia. And you have to have these kinds of organisms that do that. Most of them are on the roots of plants that are in the soils, and many bacteria, many archaea are nitrogen fixers. Life can't live without these crucial organisms, archaea and bacteria, that fix nitrogen for us. Most of them are under the soil, found on the roots, but they're nitrogen fixers. But bacteria and archaea, especially bacteria, live inside of us. We've talked about this. They're a symbiotic relationship. We can't live without them. We ingest them early on as infants, usually from the nipples of females, whether it's calves and cows, mammals especially. We simply have to have these. Some of them are pathogenic, but we have to have many of these organisms. Here are just some of the places where microbes live in our bodies: skin, mouth, eyes, scalp, large intestine, small intestine. We simply need these organisms inside of us. But you know this as well as I do. Some are very pathogenic. Some we have vaccines for. Some we do not. Some we do not. We're not talking viruses here. We're talking bacteria. In fact, when you go to the doctor sometimes and you have a cold-like symptoms, flu-like symptoms, if it's bacterial, you can get an antibiotic for it. Take an antibiotic. If it's viral, you know this. You've gone to an outpatient. You've gone to the local, outpatient, office. If it's viral, they simply say, tough it out, take some Advil. There's not very many antiviral drugs that we can take, but many bacteria are pathogenic. They enter through wounds, infections. They enter through our eyes, nose, ears, mouth. Um, some will use, remember that glycocalyx to kind of attach to a host cell and then multiply. You develop symptoms because your body is trying to reject it. But there's a little bit of work here that we use prokaryotes, especially bacteria, to break down other things. We use them in labs. We use them in industry. We use them to make drugs, especially in the pharmaceutical industry. We simply have to have these bacteria. So let me move on to just a few things. I'm going to let you kind of walk through the investigating life. Basically, it's the part of your notes that just try to bring it a little bit closer to home here, especially in the agricultural industry. Hey, that's a 31-minute lecture. I don't want to go any further than that. This is chapter 17. Remember, this is chapter 17, the bacteria and the archaea. We're going to move on to the third domain in chapter 18, which is going to be starting with the protists, the protistans. So we'll drop that probably in another day or so. Probably give you another about a 20-minute lecture here in just a couple of days.